I'm going to talk about complement fixation test. This is another type of test to prove the antigen-antibody reaction. And here you require sheep RBCs readily bound to anti-sheep RBCs. Anti-sheep RBCs can be produced by injecting the sheep RBCs into other animal like rabbit or horse or goat. The antibodies produced against the sheep RBCs can be purified and they are added to the sheep RBCs. Thereby, they react with the antigen of the sheep RBCs when remain attached to it, and they are used as indicator. So, sheep RBCs readily bound with anti-sheep RBC is used as an indicator in this test. Then you require the serum sample containing the complements. Complements are proteins of enzymatic nature, and 9 complements are there which are responsible for recognizing the antigen-antibody complex and bind to the Fc arm of the antibody and get activated, and on activation they bring about lysis of the cells. So a serum sample which readily contains active complements is very much required in this reaction. Then the serum sample to be tested for the presence of antigen or the antibody is required. Let us see how this test is performed. We'll take the first example at the top. The test sample, the serum sample to be tested for the antigen is treated with the corresponding antibody, and that antibody is supposed to bind to the antigen that you are looking for. The serum sample that is going to be tested for the antigen means presence of antigen. You have to add the corresponding antibody to that antigen to that test serum sample. And then after adding the antibody, mix it thoroughly. Afterwards, you add the serum containing the active complements. If the serum sample to be tested, which is to be tested for the antigen, does not contain the antigen, the antibodies that you added corresponding to that antigen will not be bound, they will be free. Therefore, the complement that you added will be free in the mixture. Now to that you are adding the indicator. As I had mentioned earlier, the indicator here is sheep RBCs readily bound by the antibodies against the sheep RBCs. That is what is shown here. Indicator system refers to the sheep RBCs readily bound with the anti-sheep RBC antibodies. The complement which you had added is not engaged. It is freely present in the mixture. It will immediately recognize this indicator complex. The indicator complex is now the sheep RBC readily bound by the antibody, which is nothing but it is an antigen-antibody complex. As I had mentioned earlier, the complements readily recognize the antigen-antibody complex and they get attracted to the Fc arm of the antibody. As shown in the next diagram here, the complements have gone and bound to the Fc arm of the antibody and get activated and begin the lytic reaction. The lytic reaction shown by the complement will be in such a way that the sheep RBC undergoes hemolysis and releases the hemoglobin. The entire suspension becomes red in color because of the release of the hemoglobin. Now this reaction has taken place. Why? Because the sheep RBCs were readily bound by the antibody, which is an antigen-antibody complex. The complement that you had added to the mixture was not engaged by the antibody that you had added to the serum sample to detect the antigen. Therefore, that free complement which was available in the serum readily recognized the indicator and it bound to the Fc arm of the antibody, sheep antibody, and caused the hemolysis of the indicator, that is sheep RBC. This indicates the serum sample that you are testing for the antigen does not have the antigen. We will move to the second example at the bottom. The serum sample readily contains the antigen that you are trying to detect, to which you have added the antibody corresponding to that antigen. And that antibody readily binds to that antigen. And here it is a soluble antigen, which is not visible to you. It is in the soluble form. So antibody binds to it and engages that. To that now you add the complement containing serum. Since the antigen was already there in the test serum sample, the antibody added to it got engaged and it bound to the antigens. It formed antigen-antibody complex. To that you have added the complement which readily recognizes that antigen-antibody complex and binds to the Fc arm of the antigen-antibody complex. And the complement is not available now freely in the mixture. To that mixture, if you add the indicator, that is sheep RBC readily coated or readily bound to the anti-sheep RBC, which is an antigen-antibody complex, an indicator, it will not undergo hemolysis as it had undergone in the previous case. Here, the complement is not available now to cause the hemolysis of the indicator, that is sheep RBC, because it is readily engaged by the antigen-antibody complex. Since the complement gets fixed to the Fc arm of the antigen-antibody complex, okay, binding to the antibody Fc arm, and since it gets fixed, the reaction or the test gets the name complement fixation test. And it will not be available to cause the hemolysis of the indicator, that is sheep RBC readily bound to the anti-sheep RBC antibody. So this is a test used for detection of antigen in the test sample, that is serum sample, or it could be used for the detection of antibody also. Let us presume that the test sample in which you have to detect the antibody, then you have to reverse the reaction in such a way that to bind to that antibody, you have to add the antigen, and after addition of the antigen, after mixing, you have to add the complements. If the antibody is readily there in the serum, test serum, the antigen will engage that antibody and the complement added to it will get fixed to that antigen antibody complex. And then when you add the indicator, that is sheep RBC bound to the anti-sheep RBC, it will not undergo hemolysis. It is just that we have changed the sequence. And the detection here we have done with the antigen, the presence of the antibody. Therefore, complement fixation test can be used for the detection of antigen or the antibody in the test serum sample. By using an indicator system, the indicator system here is nothing but the sheep RBC readily bound to the anti-sheep RBC, which shows positive reaction by undergoing hemolysis if the test sample is negative. If the test sample is positive, means it contains the antigen or the antibody, the sheep RBC will remain intact and there will be no hemolysis. It's a very simple test to detect the antigen-antibody complex. Thank you.